The goal of the Libertatis Ergo Holding is to advance the impact of Leiden University's activities. We do that by supporting entrepreneurs and investing in startups to bring exciting new research and innovation to the market, all to benefit society.
Today, patients are treated as numbers, and this leads to ineffective treatments with a lot of costly side effects. In the far future, there will be a personalized prescription on the treatment you need. Our MedPrint 3D printer can print with cartridges, like the coffee cups. So the pharmacy can insert the cartridges in the device, press start on the touchscreen, and the printer will make the tablets. We see that 40% of all medication for children is being adjusted at the hospital pharmacy. So the oral liquids, they taste bad. And also the children have very difficulties with swallowing these tailor-made medication. And with our printer we can make like very tiny tablets in every shape you want, on every taste you want, on every color you want. At the end of this year, the first patients will be treated with a personalized tablet.
I am really fascinated by technology and the impact it can bring to our everyday life. With Exit 71, we develop innovative technologies for the analysis of novel biomarkers, exosomes. Exosomes are these tiny particles that all cells in our body make to send each other messages. They put their own molecular information in these particles and release them in all body fluids, blood, urine, even tears. Exosomes are really the snapshots of health or disease state of organs. I don't want another great invention just to stay in the lab. And yes, somebody has to do it, somebody has to take it out, so I think I'm the right person to do this for this particular technology.
So our name Bimini is derived from the island of Bimini. It's in the Caribbean, and as the story goes, that's where the fountain of youth was supposed to be, and many explorers wanted to go there and bathe in the fountain of youth, and we want to recreate that using chemistry. We have identified small molecules that show really good results both in mice and in cell lines against cancer. Now we are taking that forward, first focus on solid cancers like colon and lung, and then also later into liquid cancers like lymphomas and leukemias. Our compounds are novel and they also target novel biological mechanisms. For instance, we are working on compounds that target the growth hormone receptor, but we are also looking at compounds that target other pathways that are strongly linked to cancer, and our molecules can modulate those pathways and then deliver an anti-cancer response. And in the next 3 to 4 years, we hope to take at least one compound towards the first in human experimentation.
Amino is a biotech company, and basically what we try to do is to identify new targets that can be exploited for immunotherapy. We want to be the best actually that can really visualize what's different on a cancer cell, and if you know what's different on a cancer cell, then you can identify what you actually want to design that therapy against. And you see a lot of companies that develop new therapeutic technologies, but we see that they develop these therapies against almost identical targets, and it's actually targets now that limited the development of successful therapies where patients can benefit from. We open the door to enable safe and effective therapies for cancer patients who are now without any treatment options.
In 2013 we were 2 guys with an idea, right? And the university stepped in and funded us. Well, in the poultry industry worldwide, 6.5 billion chicks are killed every year. Why? Well, they're male. Males don't lay eggs, so that's why we kill them right after hatching. We have a machine that's capable of genotyping the egg instead of the chick. Thereby you can choose only to hatch females. You don't have to kill the male chicks ever again. And that's better for animal welfare and efficiency. We test the eggs on day 9 of development, So our normal egg develops in 21 days into a chick, so we're relatively early. That means we think the egg doesn't really feel anything when it happens. And we take out the males and we use them for pet food actually. We have a very clear mission, right? We want to stop the killing of chicks all around the world.
We aim to make a difference in the lives of patients, ultimately, by doing what we do best, and that's building these human-relevant tissue and disease models in our chips. If you're going to look for novel therapies, novel medicines, you have a lot of drug candidates. How are you going to select which one is going to work well in a human? You have traditional cells in the bottom of a petri dish, which are actually not good enough. And you have animals, which are completely different species, so you need something in the middle. Memetas develops organs on a chip. Those are small chips, but not computer chips, but microfluidic chips, so we have tiny little channels in them. And in those channels we grow tissues from human cells in a way that they mimic as good as possible the situation in a human being. By providing these models to pharmaceutical companies, they can develop drugs that they wouldn't have been developing without us.
The name of our company is Onnes Technologies, which is derived from the great Dutch physicist Kamerlingh Onnes. So at Onnes Technologies we built a cryogenic nanopositioner system, which is a dedicated design tool for low temperature research. And low temperature research is being done at temperatures close to minus 270 degrees Celsius, which is the absolute minimum. And you need these temperatures in order to study the quantum effects, because they only occur at these very low temperatures. So typically researchers use a so-called dilution refrigerator, which not only cools down your experiment to these super low temperatures, but they also isolate your experiment, mechanically speaking, from the rest of the environment. And at Onnes Technologies we built a tool that allows you to do positioning in these dilution refrigerators while still respecting this temperature stability and vibration stability that you need in order to study these quantum mechanical effects.
I am Pauline van Deursen. I'm co-founder of Vitrochem, a company that spun off from Leiden University 2 years ago, and we are aiming to revolutionize electron microscopy. The problem with electron microscopy at this moment is that you can only look at samples that are frozen or fixated in some way. So if you want to look at biological cells, you have to freeze them first and then you can put them in the microscope. That is because the microscope is under vacuum. As soon as you put water in there, it will just disappear. Now, we are working with graphene, which is the thinnest material known on Earth at this moment. And if you put your liquid samples between 2 layers of graphene, then that forms a stable structure that is protected against the vacuum. So for the first time now we can actually look at moving samples that are still in motion. The researchers interested in these samples are biologists. They don't necessarily know how to use graphene, so our machine automates the graphene handling process so that anyone can use this technique. So this will revolutionize medical science and biology.
Bio is not a typical biotech in the sense that we are trying to discover our own medicines. Rather, we develop technologies that help to reduce the risk and improve the efficiency of the small molecule drug discovery process. And then we sell these services to our clients. The company is involved in helping to make small molecule medicines. And by small molecule, we mean the type of drugs that are usually put into a pill and taken orally, typically once a day. In the early days, most of those clients were large pharma companies like Janssen and GSK and many others. But in recent years, those companies have developed their own capabilities, having seen how important what we do is. And now the vast majority of our clients are small and even startup biotechs throughout the world.
I'm convinced that if you want to treat cancer patients, that you need to know whether the patient is responding to treatment or not. Vitroscan has great technology to test drugs in human tissues that very much are alike the tissues that are in the body. We get the tissues from the patients and we make it into little fragments with the 3D morphology. And we seed it into the assay in little wells. We expose that with the drug and then we look how the morphology changes if we put the drug on it. For example, if we look at ovarian cancer, these patients get treatment with chemotherapy and then they get surgery. But what we want to tell the doctor is whether the patient is going to respond to that chemotherapy, otherwise he can skip the chemotherapy and start with surgery. The impact on the treatment for cancer patients could be major.
The impact of having a voice disorder or stuttering severely is that people get isolated. My motivation for starting WISP is that I tend to stutter myself. And I learned, and it was kind of in the back of my mind, that when people who are stuttering severely whisper, they are much more fluent. The stutter frequency reduces with about 85%. We are developing a speech technology that is able to convert a whispered speech into a voiced speech. And it's done in real time. So you can use WISP to make phone calls, have video chats. You can use it on your mobile, but you can also use it on your desktop. We are currently in a proof of concept stage with WISP, and we are developing a first launchable product for the end of this year.